Okay.
Nice. So thank you. So I'm Vincent Moulin. I end my PhD in March, and I arrived in the Institute of Forest Ecology in RESDiNET Lab teams. And now I will make some presentation about the influence of Spruce Fungal Microbiome on attraction and resistance to Beetle Outbreaks. And we will mainly focus on condition before the attack. Okay, you will see why after. So it's I'm very happy to be in this team because it's very interdisciplinary team. I'm mainly a community ecologist and fungal ecologist and soil ecology normally. But in this team, we have people from chemistry, we have people from remote sensing, people from genetic, and people also from barbital ecology. So I think it's made a very good team to studying what is happening in a forest stand. So there is two objectives of this presentation. First one is to presenting some little state of the art on spruce bark beetle fungi association. And the second one to presenting a field experiment studying the influence of pre-attack spruce fungal microbiome on attraction and resistance to bark beetles. So we begin with this quick state of the art. So it's something that is important to consider is that both spruce and bark beetles have a microbiome, okay? It's composed of bacteria, but also fungi. And it can be categorized in different guilds on the basis of what they are doing as ecosystem function. One of the first one are the bark beetle symbiotes living on bark beetles. So N here is not the number of species of this fungi, is the number of studies that were published. I found on that guild. Okay, so many know the research on the fungi in this pathosystem have focused on the fungi on bark beetles. These fungi are carried by barbicles, and when the barbicles, arrive on the trees, then they are inoculated to the trees. And you can see, for example, it's some inoculation experiment, and you can see a dark zone near to the point that were inoculated. It's the fungus that begin to attack the tree. And when you are looking at the gallery of bark beetles, you can see the same kind of dark zone that are also fungi present on the gallery because they were inoculated here by the beetles. So beetles are vectors of these fungi that are mainly fungi belonging to the ophiostomatoid class. So this community are very important in terms of ecology because they are seeking to be weakening the tree, also detoxifying a spruce defense molecule into compounds that are not harmful for the beetles, and creating suitable gallery conditions for beetles larvae. We were also discussing something important with Luis at the coffee post because this fungi can then maybe spread to the xylem and maybe disrupt the water flow in the xylem. But I didn't so far know study about that. Maybe I need to focus to keep searching for that. Okay, so second category of beetles, they are bark beetles entomopathogens. A lot of studies are also studying these entomopathogens. They are fungi that are killing beetles. So in natural state, they will play a role in very moderately reducing bark beetle population, and several products, commercial products, have been developed with dubious to small effectiveness to reducing bark beetles. One of the main issue with these products is that it's very difficult to keep the fungi population in the stand, and you need to come again and come again and come again to inoculate population in the stand, so it's not very effective. So here you can see we have 85 studies about fungi on bark beetles. Now we will focus a little more about the fungi that are associated to the spruce, so the spruce microbiome. First, the ectomycorrhizal fungi. They are very, very important because spruce is greatly dependent of this fungi for its water and mineral uptake. It's in range 40 to 60% of mineral uptake of spruce is coming from ectomycorrhizal fungi, not directly from the roots. And in exchange, the spruce is giving 20 to 40% of its carbohydrates to the fungi. Okay, so now it's not enough from spruce, but from more general literature. Ectomycorrhizal fungi are also protecting against the root pathogen colonization and regulating the tree defense. And one thing that is very bothering me is that I didn't find study about the ectomycorrhizal influence on tree resistance during bark beetle outbreaks or even before. There's a lot of study about ectomycorrhizal fungi on spruce, but not during, not related to bark beetles outbreak. The few studies that are existing, there are studies that are only focusing on the impact of bark beetle on the ectomycorrhizal fungi. That means how fungi dies after the forest collapse. And so results are that they don't like it. Okay. Other categories of fungi, there are the root pathogens. Tomorrow it will be interesting because we are going to some field excursion in order to see some Amalia outbreak in spruce forest. There is some hypothesis that existing, but for now I didn't see recent papers that are checking if there is some synergy between Amalia fungi and bark beetles. So the idea would be that pathogen fungi that could reduce tree resistance to bark beetles outbreak. But as I was telling, the only studies that were carrying out before the rise of genetics tools. That means people were checking if there is if there is a fungal infection with microscope or visual assessment, for example, this kind of assessment. But this cannot be used in order to check for early infection of Amalia fungi. And finally, last group of fungi that are present on spruce, there are the fungi that are present in aerial tissue. That can be the bark, that can be the leaf. So if a fungi is present on a tissue is epiphyte, if it's in the tissue is endophyte. So why is that important? These fungi are present before attack, and sometimes including fungi that we also can find on bark beetles. So that means that maybe there will be this fungi will have some interest in attracting bark beetles that are the vector, for example. That these communities of bark fungi, they are disturbed by bark beetle outbreaks and descendant colonization. But study about the exact function before the outbreak are lacking. In other insect tree phytosystems, it was proven that they can affect tissue pathability by herbivores, and also consume the attacked tissue and the invader. Very recently, Kandasamy and his collaborators found that ophiostomatoid fungi present in the bark before attack can, in the laboratory, attract bark beetles in biocyte. So they make a diet experiment with where the beetle can choose between a control or a media that is colonized by some ophiostomatoid fungi, and they found that the beetles prefer to go where the ophiostomatoid is present. Also, it was associated with the production of some fungal molecules that are mainly alcohol, terpene, and esters. And they found also that this Grossmania here, this Grossmania is able to degrade spruce terpene molecule into these different compounds. Very important thing also, they found that some fungi that are not present on beetle can also produce deterrent molecules. So it's in lab, okay? That means you identify some mechanism in lab, but is not proven in field for now. So it will be very interesting to test. Last thing, just in order to have a big summary about this different study, you can see that for now research are mainly focused on beetles that are carried by on fungi that are carried by beetles and not on fungi that are carried by the spruce during the outbreak. And I will add also some things. These studies are focusing on ongoing outbreak and not condition before the outbreak. So they are not studying if fungi can be an environmental factor, that is favorizing or decreasing the risk of beetle outbreak also. So our main hypothesis, we have three main hypotheses. First hypothesis is that the bark endophyte communities that will be represented during this presentation by this are correlated with fungal volatile organic compounds before the attack. And also that trees that are attacked at the beginning or at the end of the attack will not have the same communities. The second hypothesis is that root fungal communities and tree stress before attack are also correlated. We can think that, for example, ectomycorrhizal fungi, some communities can maybe increase nutritional status of the tree before attack and reduce the risk of this tree to be attacked. And for pathogen, it will be the reverse. If a tree was deeply colonized by pathogen before attack, it will be weakened, and maybe it will increase the risk of being attacked by beetles. And last hypothesis, the fungal community differ between interior and tree age. And we know that the tree age of the stand is the place that is the most attacked by beetles. So to test this hypothesis, in April, we try to make some study. We make some study in order to describe what is happening during an outbreak. So the aim, so if you understand well, is to produce these tables here. So a volatile compound table for each tree, a fungal table for each tree, and stress indicator before attack, after attack for each tree, and ectomycorrhizal community for each tree before the attack. And we also have some additional hypotheses that we want to implement in a structural equation model. So what is a structural equation model? A structural equation model is a statistical method that is used in order to assess indirect effect, because correlation between variables, if you are looking only at direct correlation, you can miss some important mechanisms that are indirect, especially in natural ecosystems. So I will explain what there is here. So in yellow, you will find the tree distance from the age that we think is an important factor to explain difference why some trees are attacked and not and other not. Stress indicator before attack and stress and damage indicator after attack. In blue is very important. This is latent variable. Latent variable, it's a special variable that you include in a structural equation model, and it's a concept. That means it's the resultant of diverse variables. For example, tree stress before attack is a concept, okay? And all the different stress indicators will go here in order to create these variables, okay? The same with tree attractiveness here, the stress before attack and the emission by tree and by fungi. will go in order to create this concept of tree attractiveness. And we have the same with the measure density. Okay, so and you find here the fungi community that we think, as I was telling you, depends of tree distance from the edge, but also the stress of the tree before attack, mainly. And I can just add something here. The fungal community. The more important role that we have, it will not be on attractiveness of tree. It will be on not that it will be on alleviating or increasing the stress of the tree before attack. So the diverse hypotheses are set. So now we can go to the site. So it's the same site that Bashir presented you just before. So in the Sacha mountain, we have around 130 trees that were sampled before attack. Bashir had 20 trees more around that were trees that were not sampled before attack, but we add them after that in the study, okay? But only for remote sensing, not for this study, okay? We monitor the attack dynamic of beetles in 5 traps. And more about this site. So it's at the altitude of 750 meters, and the soil is very bad for spruce because it's mainly sand, it's mainly sandstone, and it's not very deep. So if you are using the world classification for soil, it's a rustic podzol, arenic, neocandic. And the humus form, they are model forms, but I need to check again if it's, I think it's this model, but I need to verify it. So there are some difference in the stand. Middle of stand and the west of the stand, there is no middle storage, only some moss, but in the west we have some Vaccinium nertigus that is covering all the understory. Okay, so in late March, beginning April, just before attack, for 33 trees, we sample volatile compounds using SPME fibers that will fix monoterpene and also esters. And it will be used then, so they will set during 20 minutes, and they will be used then to produce this matrix table. Just after that, we sample one sample of bark here, and during the three next weeks, we also sampled at 8 points on each tree barks, and we also sampled 12 samples of fruits on those. And this will be used to extract DNA, and this DNA will be used to identify which fungi are present or were present. For the beetles, we also take some beetles, and DNA from fungi on the beetles will also be extracted, amplified to identify the fungi that are present, because we think that in order to identify in our stand population, in our site, particular site, what fungi are present on the beetles, with the hypothesis that trees that are having more beetle symbiotes before attack will attract more beetles by producing more volatile compounds. And finally, here is a list of physiological indicators that will be used to compute the stress before attack and after attack. So you have this multispectral acquisition, you have dendrometer for 2 trees. You have also 2 tree course by tree that will be used in order to check for how the growth was for each tree in the 10 precedent years. You also have phloem and RNA for each tree before attack, and for RNA also after attack, and also monitoring of the trees to find when they were attacked, and a final visual assessment to check the level of the damage. So we will consider 4 groups of trees. I was telling you we have a first wave with 5 trees attack, and then the attack rise, and we are reaching at the end more than near to 20 trees, yes. So we have 4 categories: 3 killed during first attack, 3 killed during second wave attack, 3 never attack. All the 3 attacks they are at the edge, and trees in the stem interior. So now we have taken all the sample. Next step, it will be in the laboratory and then statistical analysis. So thank you for your attention.